first break? Oh,
I'll do
yay.
that. Are you seeing any changes, positive or negative, in regards to healthcare coverage with the Affordable Care Act, and whether it's easier or more difficult to get some of these expensive treatments through insurance?
That's a good question. I guess what I'll say is that a lot of it depends on the plan that the person selects. We've actually had a little bit of difficulty with the affordability of some of the, I don't want to say that, some of the better plans that some of our patients have not been able to afford the monthly payments and have chosen other plans that were more affordable. And then UT Southwestern, which is kind of contracted with most of the major carriers, sometimes isn't in the network of some of the less expensive plans, and that's been a really unfortunate aspect of care lately. This is just for pure clinic appointments, where we're essentially out of network, and then I have to find another person to help take care of those patients, and that's really difficult. So, and then with regard to coverage, I don't know, my philosophy is a lot of times we get a no, but if we fight hard enough, we'll get a yes. And so I just kind of don't take the first no for it as an answer, and just keep kind of wrangling with insurance until we get the yes that we need. And so far, we usually don't, you know, you kind of feel like you're beating your head against a wall, but eventually you can kind of break it down.
Oh, you have a second microphone. Oh, very good. Here. Borrow that for a second. Yeah, and it's challenging because it's really hard when you look at the marketplace and the plans. It's hard to understand what gets covered and what doesn't, and particularly on the drug coverage, it gets very, very challenging. I would also say that if you run into problems, call us, because we can try to help navigate as well. There's a lot of other organizations that have oncology social workers that can help people navigate, and to your point, a lot of times the physicians don't always have the bandwidth or the capacity on their staff to help individual patients go through the whole process of fighting with an insurance company or coverage. So there are other organizations that do that full time and have a lot of great assistance available for free. And then I would also say if you run into challenges for co-pays and things like that, let us know, because we can help you either get connected with a lot of the pharmaceutical companies and manufacturers have co-pay assistance programs, or there are other programs if you don't qualify for that. So it's challenging. There's a lot of things going on in Washington with regards to regulations and trying to stay on top of all of the changes in our overall healthcare system, because to your point, a lot of the insurance companies are what they call narrowing their networks. So depending upon the plan, not all of, especially an academic medical center or specialists, tend not to be covered in network, and then you end up with an additional copay, or you can't go to see the specialists. And we're trying to stay on top of all of that as well, and participate in a lot of coalitions in a bigger, broader sense to try to make sure that as these changes come down, that patients and those of us that actually have to use the healthcare system are covered, especially for a rare disease when we are outside of the norm pretty much all the time, for therapies, for physicians, for drugs, for pretty much everything. We're kind of an outlier. So I really do my best. I'll be in Washington in a couple of weeks, so we'll be participating on your behalf in Rare Disease Day at the National Institutes for Health, and then on Capitol Hill meeting with congressmen and senators about all of these different things that are going on in Washington. And then I also am participating in a pharmaceutical association meeting talking about availability of drugs and pricing and things like that. So I get the opportunity to take all of you with me as a voice and say, okay, yeah, that's really great for 80% of the population, but there's those of us over here that fall outside of the lines all the time. So what are those avenues going to be? So I would just say, we're working on it, but if you have issues, call us and see if there's something else that we can do to help. And it's gonna be a while. There's a lot of pieces and parts to our healthcare system that need a lot of work. And it'll take us it's gonna take us a while to get it all squared away. Well, that doesn't really answer your question, but call us anytime.
Thanks.
Yeah. Okay, who's next? Oh, good, one in the back.
Question for you. In 1967, my daughter Cindy was diagnosed with hepatitis. At that time, as far as I can remember, the doctor never said hepatitis C or B or whatever, just hepatitis. Could that still have been in her system and caused this disease?
So could hepatitis C be a cause of cutaneous lymphoma?
It's a good question. I'm kind of parsing through some of the,
First of all, they never said whether it was C or B. It was just hepatitis. That was an old country doctor, and that's what he said, hepatitis.
no, I could ask a few more. So, so I'll just clarify, I'll just kind of go over. So hepatitis A is a foodborne type of hepatitis, and that's a short-term hepatitis. And it's kind of like having a bad GI bug, a really bad GI bug. But if you are exposed to hepatitis A through food, essentially patients clear that infection. So, and you don't carry it. So you had it, you were exposed to it, you get really sick, and as long as you recover, then you get through it, and you don't have chronic hepatitis A. So that's hepatitis A. So that would not, and that's not known to be an oncogenic or cancer-producing hepatitis. Now, hepatitis B, people can carry with them for many years, but I'll just kind of, I can't remember the exact statistics, but let's say maybe half will kind of clear their infection, and the other half carry a chronic infection. But if you're a chronic carrier of hepatitis B, generally you might know it if it's an active form of hepatitis B, because it kind of is very hard on the liver. And people that have hepatitis B generally know when they're exposed. And if they're a chronic carrier, I mean, depends on what country you're in, but I will assume that, living in a Western country where if you're sick, as sick as you usually would be if you were exposed to hepatitis B, that someone would know that that's what you were exposed to. So hepatitis B is less common for people to carry chronically, and those who do carry it, usually they've cleared the vast majority of it. It's not known to be oncogenic. Now, hepatitis C, hepatitis B and C, if you're a chronic carrier, you can be at risk for like hepatocellular carcinomas. I guess I should say that. And then hepatitis C is something that you might or might not know that if you were exposed to it, and you can kind of become a chronic carrier. You might not have any symptoms of that exposure, and most people that are infected with it do carry it with them and don't clear the infection. The biggest cancers that one would be at risk for would be really kind of liver cancers, more so than like blood cancers. When someone has chronic hepatitis B, generally, or C, generally the cells that are kind of getting stimulated are the B cells, more so than the T cells. So, and those individuals may overproduce certain antibodies, but in terms of T cell lymphoma, I don't know of any associations. I can dig through the literature and see if there's ever anything that's been reported, but there would be no real theoretical reason that those two should be linked because of the nature of the way the disease is.
Hello. My question is, have you seen from the itching, or I don't know if it's proof of cause, but I've had a doctor tell me that there's a bruising, red, brown, patchy stuff that's grown on my
yeah.
Have you seen that? And is that, I know they told me not to research it.
Yeah. I'll explain. I can definitely explain. Okay. So amyloid just means protein. So it's a fancy medical term for protein, microscopic protein. There's lots of proteins in our body. When those proteins collect in an area, you call it amyloidosis. The real linchpin of the question is, where did the protein come from? Now, some forms of amyloid, many forms of amyloid, are from excess antibody production because someone has a internal B cell lymphoma, and those B cells are overproducing certain antibodies, and those antibodies are collecting in the skin and can cause, bruising and stuff like that. But that's because the person has a lymphoma. Okay, so that is not associated with the chronic skin itching. Okay, so at all. So in someone who has an isolated spot, when someone has an amyloid that's systemic from a B cell protein from excess antibodies, those antibodies collect in things like the tongue and around the eyes, and so people will get like a giant tongue, and they'll get bruising around their eyes, and easy bruising elsewhere on their body, but not an isolated spot. Okay? So what you're talking about is something called macular amyloid or lichen amyloid. That is because when we scratch our skin, remember that the epidermis, this top skin layer, is really just like as thick, it's as thick as a piece of tissue paper or this notebook paper here. So when you scratch your skin cells, you actually are breaking some of the keratinocytes, so you're breaking some of the skin cells. And our skin cells have proteins that give them their structural integrity. It's like the scaffolding inside of the cell. And those proteins are called keratin. And when you traumatize through scratching the skin cells, some of those proteins fall into the skin like raindrops, and they collect there. And the lichen amyloid lichen amyloidosis is basically a collection of skin protein in the skin. It has nothing to do with the cancer. It's from broken keratinocytes from a chronic state of scratching or rubbing. But the reason people tell you not to Google it is because, and even this is a concept that actually many individuals are not even taught in medical school, because you're just taught in medical school about the bad amyloid, but not this kind of like minor skin, little kind of wimpy, I don't want to say it's wimpy because it's like, just it can be quite debilitating for many patients, but when it's just in the skin, generally it's not something people die of. So in medical school, those kind of things aren't taught as much. They teach you about the things that, could really hurt somebody. And so anytime even many physicians, you say I have amyloidosis, and they're like, they think you have some kind of cancer, and you don't. So, yeah. Does that help?
I love it when people have questions.
I've read a lot about research and progress that's been made in regards to immunotherapy in the last several years. So a couple of questions on that. First of all, what's the status of upcoming immunotherapies in regards to MF? And are those generally expected to be a cure or just management similar to what we're doing now?
I think we should both answer this question. I'll take the first crack, and I'm going to hand off to Dr. Desai also, because I'm sure he has many things to add to this. So as you might imagine, when you have cutaneous lymphoma, just from the kind of little picture I showed you, there's usually a mixture of the cancerous white blood cells and some non-cancerous white blood cells, and they're kind of hanging out together because the good guy white blood cells sort of know there's some bad guys there, and they're trying to kind of get rid of them, but they don't fully. And so many of these immunotherapies are designed to kind of unleash the power of the healthy immune system. And so it's called the tumor microenvironment. And so you're kind of trying to tweak the tumor microenvironment by promoting the good guy white blood cells to fight the bad ones. actually what we're even doing when we administer things like interferon alpha or interferon gamma, because you're administering a drug that favors the kind of the good white blood cells to attack the bad ones. But I'm going to let Dr. Desai add something.
Yeah, no, I completely agree. I think immunotherapy has very broad meanings. I think when we have a blanket term means we don't really understand the process, and just like with chemotherapies or radiation for a long time, before we understood it, we knew it worked if we just threw it at a patient. I think we're still at that stage in immunotherapy, and only recently we're trying to divide up what segment of the immune system are you triggering, and what are you trying to do. So there's a immune system where you just by ramping it up, you might increase the chance of a selective attack of the body immune system against the cancer. Interferons, interleukins, all sorts of immunotherapies have been all along been doing that in a very blanket manner. More recently, there's been a lot of press for what we call checkpoint inhibitors. We know there's this immune system response, but it's being suppressed. The cancer cells express, our body grows up from a baby learning how to suppress immune system response to self. Antigens which are not expressed by self during that period of time as a child during immune system development shouldn't be allowed to exist in the body. Viruses, bacteria, cancer cells. Cancer cells have mutations. They express different antigens or proteins or dog tags, which should be recognized and killed by tumors or immune system. The process by which the tumor cells evade immune response is a very complex one, but we believe a lot has to do with these overramping of these inhibitors of that response. So by inhibiting those inhibitors, unleashing the immune system, these are the most recent wave of immunotherapy getting a lot of press in renal cell cancer, lung cancer, melanoma. I think cutaneous lymphomas have a lot of potential there. It just makes a lot of sense. We know this from other lymphomas in the body. Hodgkin's, non-Hodgkin's immunotherapies are tending to have potential benefits there. And so I think it depends on where the trial space will be for introducing those drugs. It can be tough. For mycosis fungoides, natural history is very long. Demonstrating response is very different from demonstrating efficacy or improvement in overall survival. So I think that benchmark is tough to meet at times. And so these trials, I think, are ongoing. I think the earliest, some of the most exciting checkpoint inhibitors out there in other diseases are now being trialed in mycoses in advanced stage or primary T-cell lymphomas. We don't have the response rates just yet. I think those are still ongoing. And then you add in the complexity of what I like to think about, which is we know the radiation increases the immune response, increases this blanket immune activation. So we're actually all talking about how to introduce radiation to that formula. All complex questions. The ability of immunotherapy to clear disease completely was one of your questions. There's not a strong track record there yet, but only because we're right in the infancy. So melanoma is probably where you have the best efficacy of immunotherapies at this point. Still suppresses metastatic cell. But I think you're getting response rates in diseases where heavily pretreated, refractory to every other drug, where you never had response rates before, extension survival. And I think that's just, whenever you see that, that's a generational shift in strategies. So I think there's still a lot of potential there. And we just have to figure out how to harness it and understand it better is what it comes down to. I think more than other drugs, every immunotherapy trial teaches more about the biology, even if you have a negative trial in some ways, than other drugs perhaps.
And I will also say that I'll encourage all of you, if you can get to New York in October, so we'll be holding our patient forum the 2 days after the World Congress of Cutaneous Lymphoma convenes, and hopefully we'll have a lot of those clinicians presenting and speaking about the new things coming down the pike in research in cutaneous lymphomas. What are some of those things that are being worked on to understand the biology? You know, there's kind of a couple of different things. It's in the research world, understanding the biology of cutaneous lymphomas specifically and how things are working, and then also what are the therapies and the drugs that are in clinical trials that may be targeting some of those new things that we're discovering on the biology side. So it's kind of this double-edged process, right? You have to figure out the biology and then alongside doing development of the clinical trials of the drugs to see if they're actually as effective as we think that they will be. And then eventually it'll probably be some combination therapies. What we're hearing from the scientists and the researchers is in cutaneous lymphomas, we probably will never have like a single bullet that will fix what's going on in our cells. It'll probably be a combination, but the more we learn about the biology, and as we can develop some new diagnostic tools that can tell us what's going on with your disease versus my disease, we'll be able to then hopefully target the right therapy at the right time earlier than we can do today. But we're not quite there yet, but that's the work that's being done, which I think is pretty darn exciting. Great question.
Yes.
I mean, this is just a simple one. Other than the beyond the light box, is anything else that's coming out beyond the light treatments and everything, turn to the light box,
Alternatives to
Yes.
phototherapy. Gosh, phototherapy. We love phototherapy.
I see the light box.
In terms of something that, the reasons, I'll explain why we like phototherapy. So phototherapy has been perfected over the years. Back in, we lived back at the turn of the century. You would be told to, like go to Galveston and get in the ocean, lay on the beach, because they didn't have light bulbs that, would emit certain wavelengths. But they've now done a lot of studies looking at what wavelengths are most effective at suppressing the immune reactions within the skin. And so we have sort of a UVB and UVA. UVB really has very, very low carcinogenic potential because of its limited wavelength. It's actually, if you look at kind of long-term studies, they've looked, and there really doesn't seem to be an increase in skin cancers with this form of UVB that we use now called narrowband UVB. That being said, if a patient has a history of many skin cancers or melanoma, we usually don't want to put them in anything that's UV related. And then there's UVA, which has a deeper penetration into the skin, down into the deeper layers. So when someone has, like, their mycosis fungoides goes down deep into the collagen layer, and they have maybe really thick plaques, or even maybe tumors, we would potentially use UVA because it has a deeper penetration. Also, sometimes if you have a darker complexion, more pigment in your skin, it can be harder for UVB to be effective just because it just has a harder time getting through the pigment. So now I'll explain why we like these treatments. So we like these treatments because they're very effective at suppressing the disease. And their side effect, it doesn't really affect the internal organs, it doesn't affect your cholesterol, it doesn't affect your thyroid. The biggest thing it affects is the time required to do the treatment, which is really, really inconvenient. Which is why, actually, Randy will have to talk about maybe getting your own home unit. And so, since you told your story, I can say that. What we do is once we know, the hard part is we can't just say, well, let's give you your home unit from the outset, because most times insurance plans want us to have demonstrated that UV light is effective through office-delivered phototherapy before they'll cover the unit. For some reason, phototherapy falls, for insurance purposes, falls under a different portion of the insurance plan coverage, which is it's called durable medical equipment, which is basically like if you need wheelchair, you need crutches, like you need something that you kind of always have as a tool. That's the part of the insurance plan that covers home phototherapy, and not all insurance plans cover it for this diagnosis, even though it's proven to work as one of the most effective treatments. So I don't think UVB will ever be replaced, or UVA will ever be replaced by some other thing, because that other thing would have to be something that basically had virtually no side effects, and worked really well and treated the whole body. And creams don't do that, unless you're just slathering all day, and that's not really that convenient. A cream you'd have to put it on every day, sometimes twice a day, sometimes with Targretin, 3-4 times a day. Not as convenient as potentially going to an office or in your down to sometimes once a week or once every 10 days or once every 14 days as the case may be. So I think that UV won't ever really be replaced by other things unless we find a magic bullet.
Yeah.
So
It's not really that easy because I set it up in front of the television upstairs, and I could be listening to the radio or anything, but it goes like that. And if you got the perfect situation, I mean, it's just I do it twice a week, but I'm not complaining. I used to do it 3 times, so it's great. Thank you.
thank you.
Okay.
Along the same lines, what's the average cost of a UVB unit that you could have in your home?
So they're small, it depends on the unit that one gets. The one that I most commonly prescribe is one that looks kind of like a 3-way mirror. So it's like a large panel with 2 side panels and can fold shut like a closet. There are 2 major manufacturers of that unit. There's a company called National Biological and another one called Davlin. They manufacture the same units that we use in our clinic. And for the Series 7 that Davlin makes is approximately $5,000 if someone were to buy it for cash. So it's like buying a decent used car. If you get a small little panel or a little handheld unit, like there are some that they're like a wand, and if you have like one small patch and for some reason can't use creams or whatever, sometimes doctors will prescribe a small wand, or let's say you have a small area kind of in the groin, it's hard, you don't want to go in the whole booth, you might just use the wand on that area, and those are like $500.
And there's all of the Lightbox company, the home, are on our website, and all their information and how to contact them. And I think both Davlin and National Biologics have people on their staff that can help you work through your insurance coverage and fill out the paperwork, and they're very well versed in all of that. So I always recommend folks give them a call, check out the website, and let them help you through that process. For a lot of people, especially if you live far away from a place that has a box and it's not convenient, I mean, it's great to have it at home. And you can do it as long as, you're working with your physician.
So I have B-cell supposedly attributable to my exposure to Agent Orange and Neil. So my question is, do you know of any mechanism where my condition could have induced the T-cell lymphoma in my body?
Oh gosh, I can't think of any way how that might happen. Yeah, I mean, because presumably you weren't around her when you had Agent Orange exposure on you and were touched, I guess she would have had to be. So, yeah, no, after the fact. So, these diseases are not, transmissible or catchable, and there's not something about your immune system having a B-cell lymphoma that would then induce your wife to have something in your skin that would induce your wife to have a lymphoma. Again, we don't even really know environmentally. B-cell lymphomas are a little bit different, like Agent Orange in particular is known to cause lymphomas, and certain viruses are. But with cutaneous T-cell lymphomas, right now, experts disagree. Some believe that environmental toxins, particularly things like pesticides and things that would be like an organic compound, so if you worked in like natural gas industry or something where like a lot of like noxious fume solvent type materials that would be oily or greasy, such as pesticides, that would stay on your skin for long periods, that potentially could cause changes in those like antigen producing cells or in the T cells that reside in the skin to induce a carcinogenic change. But other experts say there's been no data to really support that. it's challenging because the rates, in order to say that, what you have, statistically speaking, what you have to have is, you look at, you know what the normal rate of these cancers are, and you have to look at an area where you know everyone in that area is being exposed to the same chemical, and the rate of that cancer exceeds the normal population, and you're comparing it to the normal population. You have to do like age match controls, so you have to look at like each person who had that lymphoma, and are they the same age? Because it could, like, you know, it could be just because they're people that live by that chemical plant are older than the people who live in this other area. So, you know, you have to look at kind of matching this up, and it's really hard to do when you're talking about a really rare event like these lymphomas. So it's tough. It's a tough question to answer. But in terms of you transmitting to your wife, no.
I have another question that we just talked about. Myself too was in Vietnam and around the Agent Orange. I've got a rash on my leg that I've had all these years, but nobody's ever done anything about it or anything like that. You just brought up that there is a,
For B-cell lymphomas,
Yeah.
that's usually systemic B-cell lymphomas, like enlarged lymph nodes, coughs. So you're more internal B-cell lymphomas.
How long does it take to start seeing results from a like that?
So with light treatments, they're done, we start at a very low dose, and we have to titrate up slowly because there's what we consider a therapeutic dose, which is figured out based on the person's skin type, meaning are they very fair skinned like a redhead, or they have very dark skin, and the goal dose would be different depending on the person's native skin type. And it usually takes about 2 to 3 months to reach a therapeutic dose, treating 3 times a week. In order to titrate up, one does need to do the treatment at least 3 times a week, or really at least 2 times a week. You can't really titrate up on a weekly schedule, so you have to be doing the treatment at least twice a week. The more, if you do 3 treatments a week, you'll get to your goal dose faster than if you go only twice a week. The rate, when we see a response, it's usually around the 3 to 4 month mark that we start to see the improvement, and it can take sometimes up to 6 to see if we reach full efficacy. So I would say it usually takes us about 6 months to see how it's all gonna come out in terms of the response to treatment.
So I have a question. I think sometimes people get confused as to when should you see an oncologist versus a dermatologist. I think we're fortunate here in this area with regards to specialists and folks that know about cutaneous lymphomas. But what are your thoughts on when should you see, and what about should you see both, or how does that work?
That's a great question too. So in early stage disease, like stage 1A disease, generally that can be managed by most dermatologists. And it's always nice when patients are referred to someone who specializes in cutaneous lymphoma if one is available. But most general dermatologists will have been taught at least something about mycosis fungoides during their residency training, and will be able to manage a patient with like a limited cutaneous disease. When there's a little bit more widespread disease or difficult to control, even stage 1A, it's nice if there's a cutaneous lymphoma specialist who's taking care of those patients. In terms of involving other care, we usually don't pursue even systemic workups until there's greater than like 10% body surface area in terms of looking to see if there's anything internal in the lymph nodes, in the bloodstream, until someone has greater than 10% of their body involved. I generally will get dermatologist involved when I see patients in the tumor stage. There are a few tricks that we can pull out of our hat in just the specialty, the dermatology cutaneous lymphoma clinic, that we might try at first, and if those are unsuccessful, then we'll get the patient kind of tied in with hematology oncology for some of the other more systemic chemotherapeutic options. I think some of that is institution dependent. Some dermatologists actually write chemotherapies, but they usually have like nurse practitioners and other people who are working with them to kind of monitor those treatments. And so I think some of it depends on the person's background and their training, what their comfort levels are. But I generally like to involve hematology oncology when I know all the things that I've done are not working, and I'm sort of at the end of what my comfort level is in terms of drugs that I administer and monitor. And when someone needs more kind of chemotherapeutic treatments, then I need an oncologist kind of on board to help. And it doesn't mean that I sort of go, okay, well now you take over and I'm out of the picture. We stay in really close contact. So like Dr. Desai and I have each other's cell phone numbers, and we'll text and say, I have this patient, I need them to get in with you, and vice versa, or I've got so-and-so in clinic right now, and I have a question. So we're really like in close contact with one another about what's happening with our patients. And then many times we have patients with limited cutaneous disease, but they need something like radiation. And so I'm going to let Dr. Desai kind of talk about how he plays, what his role is.
Yeah, I mean, people generally don't see me until they've been plugged in with certainly dermatologists, and typically also medical oncologists a lot of these times. I think I do every now and then see people from outside who are only being treated by medical oncologists, and I think that's a mistake. Just because even with the systemic disease management and all things that go into that, there are just so many things about the cutaneous manifestations that affect quality of life so much more, I think, that even it's changing skin flora, what antibiotics you use, start antibiotic treatment empirically, that's outside my comfort level most of the time. And I think probably most medical oncologists say the same thing, especially if you're in a community practice where maybe you're treating one cutaneous lymphoma patient your entire, stable patients that you're treating. So I don't think I see any patients, maybe one, without you involved. So maybe, I think, yeah, very, very rarely. It's a good disease to have multiple.
Is it lunch? We're getting to lunch time. So before we break, are there any other questions that?
How common is it for the pathology on the diagnosis to be missed on one pathology for two spots, two separate biopsies on the same day?
That is actually a great question. It's a great question actually to go into lunch with, because right after lunch, I think at 1 o'clock, Scott Wickless will be talking about the pathology side. So as you recall, the diagnostic algorithm, we don't just make the diagnosis by, I might see a patient like Randy Toops and go, I'm pretty sure you've got this cutaneous lymphoma, but I don't just say, okay, I made the diagnosis, let's start this treatment. We do biopsies to kind of prove our suspicion, because the diagnosis doesn't just hinge on my clinical, it's not like psoriasis, for instance. In dermatology, we might look at a patient and say, you have psoriasis. We don't have to do a biopsy to prove that. With mycosis fungoides, it's a tricky animal, and it can mimic many, many, many different conditions. It's a great mimicker. In fact, in dermatology residency, one of the parts of our training is to look at clinical pictures and trying to be able to say, well, that is this is that, etc. And so, or to formulate a differential, like, oh, I see this rash, and it could be, these 5 different things. And one of the things that residents are taught very early on, if you're the resident who's in the spotlight in front of all of your faculty that's trying to provide differential, like list all the possibilities of the rash that's on the screen, and you're kind of, under the bright lights and on the stage, and to say, mycosis fungoides, because it can look like anything. So that's always in their back pocket. I'll just say mycosis. If I don't know anything else, I'll say it looks like mycosis fungoides, because it can really really, really mimic so many different diseases. So histologically, it also can be a great mimicker because there's normal white blood cells that live in the skin. And so after lunch, Scott Wickless will talk about the pathology diagnosis and some of the challenges and pitfalls there. So stay tuned.
Thank you.